So let's then go back to our lecture and complete the discussion of Chapter 1 and a brief discussion of Chapter 2. So we had spent some time talking about the brain and the things that it does. We have cognition, we have emotion, certainly the higher centers, the limbic subcortical areas, the hypothalamus being a key part of this axis of the higher centers and the brain stem and the spinal cord, linking with the neuroendocrine system as well. So we have the involuntary portion of the nervous system, the autonomic nervous system, and the voluntary or the somatic portion, both sensory and motor for each of those, and the endocrine systems, all integrated to be consistent with our production as an organism of an appropriate response to a change in either our external or internal or both environment or environments. So now we begin to think about, just setting my arrow here, the communication systems. So let's begin to transition away from that global understanding of organismic nervous system function and neuroendocrinology to focus on communication systems. So the nervous system uses neurotransmitters, and we'll refine all these definitions and build on our detailed, specific knowledge as we go through this semester. We know that textbooks tend to have generalities in them, especially introductory level textbooks, and at the graduate level, our task is to learn specifics. So we know that the nervous system uses more than neurotransmitters, but it's a darn good generalization. The endocrine system utilizes hormones, and that generalization is still quite solid today, but there are some additions coming into place where you notice that the immune system isn't really included in the endocrine system, and the chemicals used by the immune system to communicate aren't simply referred to as hormones, whereas if you think of the way that they work, based on the definition of a hormone, synthesized by a cell in one place, released to the body fluid compartments, and targets a distant cell, or even itself, as we will see, then that molecule is called a hormone. The cytokines do basically the same thing. So they are a signaling molecule outside cells, extracellular, and between cells, the source, the cell of origin, and the target cell that has the receptor. So each uses its own type of chemical messenger, as the general point being made in this slide. But there's a tremendous amount of interaction, as we had stated. So there's neuroendocrine interactions, neuroimmune interactions, neuroimmunoendocrine systems are even being described. We won't deal with the neuroimmuno part in this course, except just little tidbits here and there as we go along through the semester. But there are novel terminologies and disciplines in science that are emerging and quite well established now, like psychoneuroendocrinology or psychoneuroimmunology. So there is such strong correlative and causal associations between the psychic part of the nervous system or psychogenic origins of internal environmental changes, or that level of internal responses originating to external environmental inputs. So something we see hits our higher levels of psychological function first before triggering something in the hypothalamus. So the psychoneuroendocrinology and the psychoneuroimmunology are extremely important aspects of newly developing disciplines in science. So a tremendous amount of research going on in those areas. Why is it when individuals are stressed, their immune system is suppressed? And when individuals are ill, I just saw a research article in a current journal, the Journal of Neuroscience. I get the table of contents because I'm a member of the society that publishes the journal. There was an article on alterations in memory function and cognition in rats as adults if they were exposed to an infection as a fetus or a neonate. So something happened where the immune response to a pretty robust infection very early in life made a permanent change in the psychological function of the organism. The idea is to compare the impact of the cognitive versus the non-cognitive stimuli on the three systems. So the almost, if you will, the nature versus nurture aspect. What we can have in the psychogenic part of our brains is very likely to be impacted more by nurture as much as it might be by nature, whereas endocrine systems very likely are to operate under the constraints of nature, the way that we are built genetically, more so than nurture. But certainly distinctive events, traumatic events, major events biologically to an organism throughout its existence can have impact on the endocrine function. For example, if you have a pituitary tumor, that definitely changes things for the individual in the way that the endocrine systems function. But let's try to deal with this idea of non-cognitive stimuli versus cognitive stimuli with this flow chart, figure 1.2 from the textbook. So cognitive stimuli would be physical, emotional, chemical, so impacting the higher levels, cortical levels and subcortical limbic systems through the sensory system as we had described it. This information, as we know, can reach the hypothalamus. The brain and the nervous system also communicate with it. The neuroendocrine system is related to it. The non-cognitive stimuli, viruses, bacteria, tumors, through the immune system, impacting the cells of the immune system, the lymphocytes and the macrophages, releasing cytokines that have effects on those cell populations themselves or other subsets of cells in these categories, and the cytokines themselves having an effect on the anterior pituitary gland with the secretion of the ACTH. The brain and nervous system and the corticotropic releasing hormone, part of this hypothalamo-pituitary adrenal axis that we've been using as an example in the last couple of lectures. The CRH can have direct effects on the lymphocytes and the macrophage. It's now known that the hypothalamus is not the sole source of CRH. There's significant plasma levels of CRH from other cell sources, especially during pregnancy from the placenta. But within this hypothalamo-pituitary adrenal axis model, CRH not only impacting the immune cells, but the anterior pituitary to get the ACTH secretion, targeting the adrenal cortex for the glucocorticoid hormones, contributing to the tempering, at least, of the magnitude of the immune response is one way to think about it. But even more importantly, as far as homeostatic mechanisms are involved, it provides a negative feedback. So the anti-inflammatory functions of the glucocorticoid hormones serve as a negative feedback onto the immune system that has been activated through non-cognitive stimuli in this model. And obviously the classical negative feedback from the adrenal cortex, its hormonal secretion at the level of the pituitary for ACTH secretion and synthesis, and the hypothalamus for the CRH synthesis and secretion. We obviously are going to build on this regulatory component of the neuroendocrine axes as we go through the semester, but I certainly am assuming that everybody remembers that type of negative feedback for this general introductory discussion. So how do cells communicate with each other? So they're using chemical messengers, as we know. They have biophysical properties. They're either going to be water soluble or lipid soluble. Our discussion of the cursory discussion of the cytoplasmic membrane included the statement that the lipid soluble chemical messengers can diffuse through the lipid bilayer of the membrane and seek out its receptor inside the cell. So that we already see, based upon its the biophysical properties of the glucocorticoid, for example, its mechanism of action is going to be very different than a water-soluble hormone such as the CRH or the ACTH that cannot slip right through the lipid bilayer. So it's going to bounce around and bump into the surface of the cell if it reaches its receptor and hits it just right. The lock and key theory, as you know, comes into play, and you get the ligation of the receptor by the cognate ligand. So the ligation, we know we put a ligature on a cut or incision that we might have to tie it together, pull two things together. So ligation of a receptor means that its chemical messenger is bound to it. That chemical messenger that binds to it is the ligand. The term cognate means that, for example, if it's serotonin that's the chemical messenger and the serotonin receptor is involved, then that's the cognate ligand because it belongs to that receptor. That's the cognate receptor for this ligand as well. So we'll hear that term of cognate receptor and cognate chemical messenger as we go into the future. Just want to introduce it at this point. So the water-soluble molecules have to act on the surface of the cells. So there are two types of chemical messengers, the intercellular, between cells, which are also the extracellular. They're outside the cell in the plasma or interstitial fluid, the two subcompartments of the extracellular fluid compartment. So it's traveling between cells and it is outside the cells versus the intracellular. So there's going to be a large number of intracellular chemical signaling mechanisms that we will evaluate through the semester, and they are very, very important. They're branching and highly diversified. They can converge and diverge and have crosstalk between them. So it's a very complex mechanism of intracellular chemical signaling that is occurring at any one point in time in almost any cell within our body. So now we just continue on with the basics, the methods of communication between the cells. There can be endocrine, paracrine, autocrine, neuroendocrine, and neurocrine, and we'll define and describe each of these with these forthcoming illustrations. So the first is obviously the typical type of hormone function that we think of as an endocrine function. So basically there is a cell of synthesis and release, the cell of origin, the source cell for this molecule that is secreted into the interstitial fluid compartment through a mechanism referred to, as we know, as exocytosis. It exits to the outside of the cell into the interstitial fluid, generating a local high concentration. The plasma, capillary, is going to be nearby. Any endocrine tissue or endocrine organ is going to be highly vascularized, very rich capillary beds, because you have to keep the blood flowing through there, flushing out any hormones that enter the plasma and sustaining this concentration gradient from interstitial fluid into the plasma. So it's high to low, the typical diffusion that we know about. Movement from high concentration to low concentration. If it had to diffuse great distances, you'd have to generate a very high concentration locally near the cell of origin to be able to push it the great distance. So there's always going to be a capillary very close by, so a short diffusion distance to get down this concentration gradient. The plasma washes it away for whatever distance it might be, but it is distant. It is a remote distribution. They make it look like the cell might be nearby. There might be instances where that's true, but what we're more familiar with is between the cell of origin and the body cells, we're going to go, let's say this endocrine cell is in the anterior pituitary, and we've got to get to the adrenal cortex. Well, with venous blood back to the right heart, the right atrium, right ventricle, through the lungs, right? Pulmonary veins to the left atrium, left ventricle, out through the aorta, finally down to the abdominal aorta, through the renal arteries, the adrenal arteries, and we're finally at the adrenal glands. So in that example, this little arrow here has a lot built into it. So we need to keep that in mind. And I try to remind us as we go throughout. You know, it's not this simplified thing of just boom, I go from here to there. It's really sophisticated. So to sustain a plasma level, baseline level, this endocrine cell's got to be going pretty good. To increase plasma levels, this endocrine cell, as well as its buddies that are expressing the same gene for the same secretory molecule, have to be coordinated in their efforts to dump enough of this out to produce an increased plasma level so that the target cells realize there's a change above baseline and respond appropriately. So it's a cell of origin, remote from the target cell, and the interstitial fluid, plasma, and then interstitial fluid being the fluid compartments of transport. Target cells are only those that express the gene for the receptor and insert the receptor and couple it with the appropriate functional response mechanisms. We extend that simple definition that the cell with the receptor is the target cell. It has to be functional. A cell without a receptor, you're not going to get a response. And, of course, only with the receptor will you get the response. So that's a very complex description of endocrine type of communication between cells. So there is the simple definition that the source of the hormone is distant from the target cell, and the extracellular fluid is utilized as the transport mechanism to get from origin to target. Now, the local interstitial fluid does increase in concentration and diffuses farther away from the cell, doesn't just go straight to the capillary, gets out in the interstitial fluid and it can diffuse wherever it can, right? And if it gets close to a capillary, it's going to cross it into the plasma down the concentration gradient. But while it's out there, while the hormone is out there in the interstitial fluid, it's bouncing into everything. Remember, Brownian motion, the bulk flow of interstitial fluid is moving these molecules around, and they're constantly bumping into just about everything. This is where my name comes in. They collide with everything, right? So we keep that in mind. So if the cell secreting the hormone expresses the gene for the receptor of its own hormone, then it can impact itself. That is called autocrine. We'll call this autoinhibition when we talk about some of the neurotransmitters. And typically that's what it is. If too much extracellular fluid concentration is there, it's not diffusing away. It seems that the cells, many endocrine cells and many neurons, have devised a method to signal back to themselves and say, you're wasting hormone, you're wasting transmitter, don't dump it out there. Here we're specifically thinking about hormones, but we'll apply this term as we get more detail. If it just targets neighboring cells, regardless of what these cells might be doing, and what their response might be, if they're nearby, then that's referred to as paracrine. So it's still the extracellular fluid that is carrying the extracellular chemical messenger, but it's a shorter distance, and the plasma typically is not involved. It's just the interstitial fluid portion of the extracellular fluid compartment. So still a cell of origin, the source. It's a distant target cell, but not too distant. And it doesn't involve the plasma part of the extracellular fluid. It only involves the interstitial fluid to deliver. And that, of course, is almost a similar description for the autocrine, except it doesn't travel very far beyond the cell of origin to provide a negative feedback, or a feedback at least. Typically, it's a negative feedback as an autocrine function. So I know these aren't complicated concepts, but I'm trying to throw in a bunch of other words to make you think about the levels of sophistication that are important, obviously, at a graduate level course. And we start digging into the literature. If we don't think of these specifics, the complexities, then we tend to have simplistic models like these illustrations in our head, and we're easily convinced by authors to view their conclusions as definite, and we're less likely to question them. So that's the idea of this course, is start off with these generalities, the introductory types of ideas and concepts, and layer on complexities as we go along, and develop the skills to question textbooks, question review articles, and question information being presented to us in primary research articles. So hopefully that helps you get through these lectures. So now we're adding the neuroendocrine part of it, where there is a neurohormone. So the neuron is now the cell of origin, and we'll discuss synthesis and secretion, but basically neurons are highly specialized secretory cells. And when it gets right down to it, the site of release, in this case the axon terminal, the mechanism is the same: calcium-dependent exocytosis. Now, exactly how the calcium channels get open to let the calcium in might be different than a pituitary cell, but when it comes down to it, you've got to have calcium, you've got to have all the molecules required for exocytosis, the SNAP and all those other things that you might be familiar with and that we'll review in a cursory fashion for this course. And the vesicle has to undergo the appropriate type of binding, everything for exocytosis that we will discuss in a bit more detail as we proceed through the semester. So again, it produces a local interstitial fluid concentration that is of higher concentration there than in the plasma. And again, where we have this type of a junction between a neuron axon terminal and a capillary bed, it is called a neurohemal junction, H-E-M-A-L. So neurohemal junction is neuron ending near hema, or blood, a capillary. So again, not a great distance between the site of release, the localized high interstitial fluid concentration, and highly vascularized tissue to wash away whatever gets in the plasma and sustain that concentration gradient. So that or virtually all of, if you want to think of it that way, it's never all of it, and probably not even virtually all of it, but most of what is released gets washed away in the blood, because that's the whole idea here. That's the functional system that has evolved, let's term it that way. Some of these molecules can escape, can have autocrine, maybe paracrine functions, but the intention is, of course, we know biology doesn't have intent, but it's hard not to use that word sometimes. The intention is to get it into the plasma, wash it away to a remote target cell, and again, only the cells with the receptors can respond. The neurocrine type of signaling is shown in this slide. Let me just go back quickly to the list there. So endocrine, paracrine, autocrine, neuroendocrine, and now neurocrine. Okay. So the neurocrine is neuron directly onto a target cell. This target cell can be virtually whatever type of cell it is. It might be another neuron. It might be another type of cell that would respond differently, like a smooth muscle. So this is the one we are most familiar with if we've studied any kind of neuroscience. The neurotransmitter is the chemical messenger, and of course it's electrical signal, but the mechanism for release, again, exocytosis. So we know the neuron is a specialized secretory cell, still uses exocytosis. What really makes it special is it couples the electrical signal, the action potential as we know, to the exocytotic event, whereas release from an anterior pituitary cell might be through transmembrane signal transduction mechanism triggering an intracellular chemical messenger that brings the calcium to the site where it's needed to cause the exocytosis. Still, calcium, exocytosis, so on and so forth. So we'll take a look at one research article when we talk about the exocytotic secretion on how that function is conserved even from the level of yeast cells. So some of these basic mechanisms are conserved over vast distances between species and genus and stuff like that. So I'm going to take some time with this slide to go through all of these examples, and this is that stage where we do that type of thing, and then we look at specifics later. So this slide is taking us through 6 examples of neuroendocrine or neurocrine, and I won't categorize them as neuroendocrine or neurocrine. I'll give examples, and we'll see how we'll study these as we go along. First, for the key, we see the S is the stimulus, the R is the receptor on some type of afferent neuron using a neurotransmitter. Within the integrating center, there might be many integrating neurons here, reaching a final common pathway for an output of the integrating center. The central nervous system is a prime example. The efferent neuron will use a neurotransmitter in this case, or in other cases, a neurohormone. These illustrations are different. The circle is a little bit tinier for a neurotransmitter versus the neurohormone. Classic hormone is shown as a square, for example, here. And then the target cell. So this example is, let's say, a pinprick on your fingertip. Receptor brings that pain information to the spinal cord. You get the monosynaptic reflex to the appropriate skeletal muscle to pull your finger away, and we know information in the integrating center is going to go up, and you might even say, ouch. But this is a typical type of spinal cord reflex that we're illustrating here. So only neurotransmitters involved. The second example, stimulus to receptor to neuron, neurotransmitter in the integrating center, but a neurohormone being released into the plasma. So this could be an example of oxytocin secretion, mechanical stimulus to the nipple, carrying the sensory information through the integrating system up to the hypothalamus, activating or increasing the number of action potentials of the oxytocin neuron. Oxytocin is released to the posterior pituitary, reaches the appropriate target cell in the breast, and contributes to the milk letdown reflex. Third example, stimulus, receptor, neuron, neurotransmitter in the integrating center, endocrine cell, neurohormone onto an endocrine cell with a hormone targeting distributing through the plasma to the target tissue within the target cell within the target tissue. This example could be prolactin, although I'm not really sure what the neurostimulus would be. But anyway, from this point on, could be neuroendocrine hypothalamus, affecting anterior pituitary release of prolactin that would target the breast tissue as well. I don't know exactly. It would have to be some type of hormone that would be the stimulus here, but that's not the best example. Then the fourth example is the same, except we now insert a capillary plasma delivery system between the efferent neuron and the first target cell. Oh, this wouldn't work as prolactin because this would be the prolactin here, because from the hypothalamus to the pituitary for prolactin release, a capillary bed is included. Oh, this one's the adrenal medulla. Yeah, it's coming back to me now. So this would be a preganglionic sympathetic neuron in the spinal cord, goes out, innervates the adrenal medullary chromaffin cells. We'll talk about how the acetylcholine released by this neuron targets the nicotinic cholinergic receptor on the adrenal medullary cells and brings about the secretion of the adrenaline or epinephrine that goes into the plasma to target the target cells within the target organs, for example, the cardiac myocytes of the heart to increase heart rate. Okay, so this one is the prolactin. Number 4 is the prolactin with a capillary bed into hormone between the hypothalamus and the pituitary. And then number 5 is the more classical type of neuroendocrine axes that we'll talk about. For example, the hypothalamo-pituitary adrenal axis, where we have stress coming in through the afferents integrating center onto the CRH neuron, CRH into the interstitial fluid, washed away down to the anterior pituitary, binding to the receptor, stimulating synthesis and secretion of ACTH. ACTH secreted into the interstitial fluid, washed away in the capillary bed, off to the heart, off to the lungs, back to the heart, through the aorta, the adrenal glands finally receiving the ACTH where it'll bind to the receptor and bring about the release of the ACTH. ACTH reaching the adrenal gland, binding to the receptor, and causing the secretion of glucocorticoids, got the wrong hormone there, from the adrenal gland, and the adrenal corticoids reaching the plasma to target the target cells. And then finally, the stimulus can be directly at the level of the endocrine cell, such as the pancreas, but there can also be a neuroendocrine cell inserted here, such as the osmotic stimuli that affect the vasopressin secretion from the hypothalamus. So this would have to be slightly modified to reflect vasopressin secretion, but the stimulus can be directly on the source of the hormone, whether it's neuroendocrine or endocrine, and bring about the release in interstitial fluid with it reaching the plasma to be delivered to the target cells. Thus, we can proceed to the quick discussion of the types of chemical messengers. There's phytohormones, such as gibberellins and phytoestrogens. There are parahormones, the histamines and prostaglandins, prohormones that we've mentioned, growth factors, cytokines, vitamins, true hormones, neurohormones, neurotransmitters, pheromones. And the question is, are phytoestrogens pheromones? So let's define some of these, and then we can answer that question. So a pheromone is a volatile chemical messenger, or they are volatile chemical messengers. There's classes of them, synthesized by exocrine glands. So they exit the organism, actually. So they're not endocrine hormones. They exit into the external environment, secreted into the external environment. They act on other individuals, usually in the same species, and typically through the olfactory, smell, or gustatory or taste receptors. And they alter behavior, called the releasing effects, or through what are called primer effects, they can affect the neuroendocrine system of the target organism. So the question before was whether phytoestrogens, that it's a hot over-the-counter sales item for supposed treatment of, I don't know if there's any, I think there's some clinical evidence, but anyway, it's pushed more so as over-the-counter, non-prescribed treatment of menopause to take phytoestrogens such as from the soybean. I think we're all aware of that. So if we consume plants that have phytoestrogens in them, they are not acting as pheromones in us. They're actually acting as an agonist, an exogenous agonist to bind to our hormone receptors. And that doesn't fit the definition of pheromone at all, because we are not in the same species. We're not a plant, right? So hormones then are classically defined as chemical messengers effective in small quantities. So this is very important. Picograms per ml, nanograms per ml. So quite small quantities, but potent biological effects and responses to cellular, organ systems, and organismic levels. Synthesized in ductless or endocrine glands, so they're secreted into the interstitial fluid, then delivered by the circulatory system. I leave this definition on here because it's the one that we classically see, but we now know that it's wrong. They're secreted into the interstitial fluid and then transported through the circulatory system to remote target cells. Acts on receptors on specific target cells located at a distance from the site of synthesis, exerts a specific physiological or biochemical regulatory action on the target cell. We now can begin to take into consideration other terminology that has had to emerge to explain less than classical phenomena as more and more research has been performed on these chemical messengers. So neuroregulators can be either a neurotransmitter or a neuromodulator. And there are a variety of definitions of these. I typically go with neurotransmitters being the fast-acting molecules, neuromodulators taking more time, but also can be neurotransmitters of being a gas pedal where you're either putting the gas pedal on or a brake, putting the brake on, with short-acting effects, so excitation and inhibition, the short-acting ones, and neuromodulators having slower effects, and just as we had described it before, not so much an on-off switch, but the rate of coming on, the rate and extent of the response, so how many action potentials will you get from one excitation, and a wide variety of things like that that I think we're best to leave until we discuss it in detail. But these can be the light switch on-off, if you will, neuromodulator being the rheostat that we turn to make it more dark or less dark. Then neuropeptides are included in neuroregulators. Very often they are neuromodulators. But they can act as hormones or non-hormone peptides, and they can, as I stated, typically be neuromodulators. And they're considered those peptide neuromodulators that are synthesized and released by neurons. So an example is cholecystokinin, when produced and released by a neuron, is considered to be a neuropeptide. But when produced and released by the pancreas, it's a pancreatic hormone. So we have tables in our textbook that go on and on with all of these. 1.2, the neuroregulators, neurotransmitters, and neuromodulators, and neuropeptides, and these are defined. We are out of time at present, so I will do a separate lecture to go over this table and then complete the discussion of Chapter 2 on Friday, and we will probably proceed with the Chapter 3 as well. Thank you for your time and attention.